Welcome back to another Edexcel Further Additional Science video. This is part of Biology Topic 1 and it's about vaccines and antibodies. So, vaccines. What was the history of the vaccine? Well, Edward Jenner, this is how it first started. I always remember the Jenner as Edward Jenner as the Jenners. He gave a pathogen to a boy and that caused cowpox. So the boy developed the cowpox and then recovered. And then he gave him another pathogen which caused smallpox, but he didn't develop it because the vaccine, the pathogen that he'd given to him previously for cowpox, because cowpox and smallpox are very similar. Once the lymphocytes had killed the cowpox, memory lymphocytes that could do the same thing for smallpox were still in the bloodstream and they could attack the smallpox. So they did not develop any symptoms from smallpox. So, vaccines is basically the immunization process. So first off, a vaccine containing a dead or weakened pathogen is injected into the body, and it contains antigens, which are horrible. It must be dead or weakened because otherwise the lymphocytes cannot attack it very easily and they have to do a lot more work. So if it's strong and healthy, it's just going to cause the disease. It has to be dead or weakened so that the lymphocytes can just attack it and get familiar with it. And then all the lymphocytes that it creates around this pathogen are fitted with antibodies that fit perfectly into the antigen. It's kind of like the lock and key theory with enzymes. There's every enzyme is perfectly shaped to fit into the docking. So it's kind of like that. You would have done that in additional science. So the lymphocyte divides over and over again to produce more identical lymphocytes that will do the same job. Some of these lymphocytes will produce large amount of antibodies and kill the pathogen, but some of them won't be used up, so they will remain in the bloodstream as memory lymphocytes in the case that the pathogen re-enters the body so it can attack it and they won't get the symptoms from it. That's immunization in a nutshell. So what are the advantages and risks? So all medical procedures have risks, and so do vaccinations, although they're not very dangerous risks. The risks are that some people might get mild reaction, swelling or soreness, and it's very rare that a person would develop major harmful reactions. And the advantages, as you can see, far outweigh the risks here. Immunity is produced without being ill, so that's a major advantage. Immunity lasts for a long time. Sometimes you might need to get boosters. People get their jabs when they're babies, and then for tetanus they get them when they're in year 10, I think it is. I think I had mine last year. Not fun. And then if most people are immune, then the few people that are not immune are less likely to catch the disease because it's less common. And once the majority of the population are immune to these diseases, they just go away. Like cowpox and smallpox aren't really a thing anymore. Sometimes they are in less developed countries though. So, antibodies. This is a little bit about response times. If you look in the revision guide, there's a nice graph for this. So, after the first injection with the pathogen, it takes about 10 days to produce enough antibodies to attack it. That's why we feel quite ill for a while. And then once it's done, it takes about 10 days to kill off the virus or whatever it is that you have, common cold, whatever. It takes about 10 days to do that, and that's called the primary response. And then say if you injected the same pathogen about 100 days, the secondary response takes less time, and as there is already memory lymphocytes in the bloodstream that can attack the same disease. And it works a lot quicker as well, so you don't feel ill, or if you feel ill at all, you don't feel ill for a very long time. Now, monoclonal antibodies are antibodies that can carry chemical markers or treatments, like drugs. Monoclonal antibodies are identical as they are produced in large quantities by hybridoma cells. So, You're probably thinking, what are hybridoma cells now? So how is a hybridoma cell formed? We'll go into what they do in a minute. So you take a B lymphocyte from a mouse, and that fuses with a cancer cell to form a hybridoma. Now, hybridoma cells divide and produce monoclonal antibodies, which are all the same. So, what are the added advantages and disadvantages of B lymphocytes and cancer cells? So the B lymphocytes, they make particular antibodies, and they make them continuously, so they don't stop making them, they always make them. Disadvantage is that they don't divide. So this is how you see they come in really well. They're a really good pair with the cancer cells, because the cancer cells divide continuously, which obviously the B lymphocyte, which I just said, don't. And the cancer cells can be channeled to attack certain cells. The disadvantage is that they don't make antibodies, which B lymphocytes do. So they're the disadvantages, and they fit the advantages and disadvantages work well with each other, which is why they are such a good pair. And once they come together, they fuse together, they make a hybridoma cell, which produces monoclonal antibodies. So what can monoclonal antibodies be used for? Well, they can be used in pregnancy tests to test for the hormone in urine. So when you are pregnant, there is a hormone that is released, and that can be found in your urine. So those little sticks will contain monoclonal antibodies that will detect the hormone when you pee on the stick. And then it will tell you whether you are pregnant or not. But they're not always very reliable. They can also stick to blood clots or cancer cells to detect them or to treat them. Like I said, they can be channeled to attack certain cells because of the cancer cell. So what they are also used in radiotherapy and drug treatments, but they can't be targeted precisely for that, and can damage any other cell in the body. Monoclonal antibodies can also have drugs attached to them by doctors to deliver them to specific cells. Alright, and that's it. If you have any questions, please do comment down below. I will endeavour to answer them for you.